The central dogma begins with DNA replication. DNA consists of a deoxyribose sugar, phosphate group, and nitrogenous bases, including adenine, thymine, guanine, and cytosine. Adenine and guanine are purines, which have 2 rings, and guanine and cytosine are pyrimidines, which have 1 ring. DNA is directional in both strands, indicated by the 5' and 3' end. The 5' end has a phosphate group attached, while the 3' end has a hydroxyl group attached. This directionality is important for replication as it only progresses in the 5' to 3' direction. However, the replication fork is bidirectional, meaning one strand is oriented in the 3' to 5' direction, which is the leading strand, while the other is oriented into 5' to 3', which is the lagging strand. DNA replication occurs in different places for prokaryotes and eukaryotes. In prokaryotes, it occurs within the cytoplasm, whereas in eukaryotes, it occurs inside of the nucleus. DNA replication is a semi-conservative process, meaning that each strand in the double helix acts as a template for synthesis of new complementary strand, resulting in DNA molecule with 1 original strand and 1 newly synthesized strand. The first step of DNA replication is the unwinding of the 2 parental strands, in which the base pairs are broken. These bonds are broken with the help of an enzyme called DNA helicase, which disrupts the hydrogen bonding between the base pairs in order to separate the strands into a Y shape called the replication fork. The topoisomerase enzyme also aids in the process by providing stress relief to untangle replicating DNA. The second step is the binding of a primer. Primer is a short piece of RNA that is generated by the enzyme DNA primase. Primer binds to the 3' end of the leading strand once the DNA strands have been separated. The primer always binds as the starting point for replication. RNA nucleotides are found in the DNA replication in the RNA primer, which initiates DNA synthesis, as primers are required for DNA synthesis because no known DNA polymerase is able to initiate polynucleotide synthesis. The third step in DNA replication is elongation, in which DNA polymerase enzymes create a new strand. This occurs when DNA polymerase binds to the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication. This newly formed strand is continuous because replication proceeds in the 5' to 3' direction on the leading strand. The lagging strand begins replication by binding with multiple primers, each of which are only several bases apart. DNA polymerase then adds a piece of DNA called Okazaki fragments to the strand between primers. This process, however, is discontinuous because the newly synthesized fragments are disjointed. In this step, DNA polymerase 3 is the main enzyme catalyzing the 5' to 3' polymerization of DNA strand during replication, whereas DNA polymerase 1 is the main enzyme for repair, removal of primers, and filling the gaps in the lagging strand. The fourth and final step to DNA replication is termination. Once the continuous and discontinuous strands are formed, an exonuclease enzyme removes the primers from the original strands, which are then replaced with bases. Another exonuclease enzyme then proofreads the newly synthesized DNA in order to remove and replace any errors. At this point, DNA ligase joins Okazaki fragments together to form a unified single strand. Then a type of DNA polymerase called telomerase catalyzes the synthesis of telomere sequences at the end of DNA, which act as protective caps to prevent nearby chromosome fusing. Lastly, the parent strand and its complementary DNA strand coils into the double helix shape, resulting in 2 DNA molecules, each containing 1 strand from the parent molecule and 1 newly synthesized strand. The 3 types of mutations that can affect DNA replication are base substitutions, insertions, and deletions. Base substitutions essentially swap different nucleotides for one another, meaning that only one nucleotide within a gene sequence is changed, so only one codon is affected. Insertion affects the DNA sequence by adding 1 or more nucleotides to the gene, which may result in the dysfunction of a protein. Deletion affects the DNA sequence by removing at least 1 or more nucleotides in the genes, which may also lead to the dysfunction of a protein. The second step of the central dogma is transcription, which marks the passing of genetic information from DNA to RNA. In this process, a gene's DNA sequence is copied to make RNA. RNA is very similar to DNA, however, it has a ribose sugar rather than deoxyribose, which is an additional oxygen present, and contains uracil rather than thymine. There's also a phosphate group and nitrogenous bases. The main enzyme involved in transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. It has 3 stages: initiation, elongation, and termination.
The first step is initiation. In this step, the enzyme RNA polymerase binds to a sequence of DNA called the promoter, found near the beginning of a gene. Binding to this promoter initiates the transcription process. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template that's needed for transcription. The enzyme helicase also plays a role in this process. The second step is elongation. One strand of the separated DNA acts as a template strand for RNA polymerase. As the enzyme reads the strand one nucleotide at a time, it builds an RNA molecule out of complementary bases from the 5' end to the 3' end. The RNA transcript carries the same information as the non-template coding strand of DNA, but it contains the base uracil instead of thymine. RNA polymerase adds nucleotides like adenine and uracil and cytosine and guanine, which are paired respectively. The third and final step of transcription is termination. In this step, sequences called terminators in the DNA signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase as an individual strand of RNA. After this process, the copied RNA acts as mRNA, or messenger RNA. In bacteria and other prokaryotes, it can become an mRNA right away, but in eukaryotic cells, pre-mRNAs must have their ends modified by the addition of a 5 cap at the beginning and a 3 poly A tail at the end. Many also undergo splicing, where parts of the pre-mRNA, introns, are chopped out and the remaining pieces, called exons, are stuck back together. This process increases genetic diversity by modifying the types of proteins and their amounts that are produced by the mRNA. This process is similar in prokaryotes and eukaryotes, but there are some key differences, besides just the addition at the end. Eukaryotic transcription takes place inside the nucleus, while prokaryotic transcription takes place in the cytoplasm. This is because in eukaryotes, transcription and translation are spatially separated. In eukaryotic transcription as well, proteins known as transcription factors are also needed, while in prokaryotic transcription, they aren't.
The last step of the central dogma is translation, where the mRNA is transported out of the nucleus into the ribosome, also known as the factory for protein synthesis. In this project, the cell uses a type of RNA called transfer RNA, or tRNA, to help assist with the journey. Ribosomal RNA plays a role in protein synthesis by ensuring that the codon sequence of the mRNA is translated. In both eukaryotic and prokaryotic cells, translation occurs in ribosomes in the cytoplasm. Each mRNA is a triplet with 3 bases known as codons. Codons contain the genetic coding and information for specific groups of amino acids, which are later used by the ribosome to generate them. As the messenger RNA passes through the ribosome, each of these codons matches with the corresponding anticodon of a transfer RNA proton through a process known as Watson-Crick basis pairing, which is the common adenine, thymine, and guanine cytosine detailed earlier. Carrying an amino acid as its 3 terminus, the tRNA molecule is later incorporated into the growing protein chain, ultimately being expelled from the ribosome. There are many different types of tRNA, each of which can read a couple codons and bring the right amino acids that correspond with these codons. To do so, all of them have 3 slots, the A, P, and E sites. They can also act as an enzyme, helping to catalyze the chemical reaction that links amino acids together to make a chain. All of these resulting polypeptides or proteins are built at the ribosomes. All of them also have 2 units, the large and small, which both come together to surround the mRNA and help ease the transformation.